Thank you so much, by the way, for having your time to interview with us.
Of course, always.
Okay. All right. Thank you so much. All right. Okay, I will get it started. So yes, hello everyone. My name is Ucher Jung, the CEO and President of GSEO, and I'm here today to interview Emma Delgado, a senior at American Heritage Schools. Despite being in high school, Emma has completed two research papers and is currently working on her third. She has acted in research competitions, notably qualifying for ISEF, or International Science and Engineering Fair, twice in a row and winning in her 2023 competition. Her work has also been published in the Journal of Student Research and featured on Junior Edison TV. Yes, this incredible person is here with us today. Without further ado, shall we dig and learn more about her? So yes, Emma, could you briefly introduce yourself to our audience?
Emma Delgado. I'm a current senior at American Heritage, so that means I'm graduating this year. I'm involved in science research, of course, and my main focus in my research is Alzheimer's disease. So I'm looking for preventative mechanisms in regard to Alzheimer's. I'm also active in my school newspaper and also in my school's business club and Model UN organization.
All right, that sounds fantastic. And our first section of the interview will focus more on your research papers and your research experience. So while I was exploring your LinkedIn page, I was able to find that you have completed and is currently in the process of completing three research papers with the Summer Science Research Program. So could you please describe more about what that research program does and also explain about how you found this opportunity?
Yeah, so actually, my school has offered this science research program, and I decided to join in the beginning of my sophomore year, going in the summer before my sophomore year, and I really just wanted to find a passion project. So that's why I decided to join the science research program that my school has. And at this program, we're able to pick a topic that's interesting to us and then delve into research and research that topic specifically. We're given a budget of about $1,000, and then we
Oh
can use
wow.
that buy materials that we will need for our experiment. There is a fee of $500 to pay, but we get that in return with our $1,000 budget. And then
Oh,
we also are able to just work in the lab over the summer and also during the fall after school hours. So I was in the lab
okay.
about maybe for maybe June, July, and then also every single day after school from about 3 to 6 p.m. to work on my research.
Oh wow, this sounds actually fantastic. A great support from your school, actually. Yes, your three research papers were all about Alzheimer's disease, as you mentioned. So first, how did you find this interest in this area of the field? And can you discuss more about your journey to narrow down on this research paper topic?
Yeah, for sure. So, I knew when I joined science research that I wanted to research Alzheimer's just because I've experienced it firsthand with my grandfather. He had Alzheimer's before he passed away, so I wanted to research into that, as well as my grandmother's have it. So, it's something that's really close to me in my heart. So, I wanted to research it to maybe find a preventative mechanism so it doesn't affect my family or my mom or my sister. So that's how I knew I wanted to do Alzheimer's, and then to narrow it down to a specific topic. Well, my first year project, I wanted to do something that was more non-invasive because my grandparents were kind of like they weren't a big fan of like medicine and needles and all that. So I tried to find a more non-invasive approach for Alzheimer's disease. And then my second year project, I wanted to look into the gender discrepancy actually because females are two-thirds more likely to be diagnosed with Alzheimer's than males. And
Mm.
who has a younger sister and who is also has the genetics of Alzheimer's in her own bloodline, I wanted to see if there were any preventative mechanisms that I could take.
All right, that sounds interesting. And what I could find from your LinkedIn profile also is that you worked on this research paper since grade nine. And for grade nine, of course, Alzheimer's disease is a very challenging topic, of course. So I believe that your journey must have been very tough and challenging. So was there any motivation that stood you up despite all this, challenges that you might have faced?
Yeah, so I mean, I mainly wanted to join science research in the beginning just to complete a passion project, to have something where I could just say, yes, this is mine, this is my own, and have no one else like be able to be like, I helped contribute, I helped contribute because this is like solely what I'm passionate about, and that's mainly why I wanted to just complete research. And then as I started to
Mhm.
complete research, I realized that I really just loved research and I wanted to continue
Oh,
with it,
okay.
one passion project, but do a couple more.
All right, sounds interesting. So, yeah, you might have worked on at the lab, I believe. So, was there any like challenging experience while conducting your research paper, and how did you overcome those challenges?
Yeah, so for my first research experiment, I actually had to learn how to shock my flies to test their long term memory responses. And that was sort of a challenge because I didn't really know how to conduct that shocking. And in the beginning, I sort of accidentally like used a really high conductor of electricity in the lab, and and then I put an open circuit into that high conductor of
Okay.
electricity, and that caused sort of a negative reaction in the lab, and smoke started coming out of the machine. But we
Okay.
figured that out. But that was one of my main issues when I first started researching
Mhm.
this topic. But we figured it out in the end.
Interesting, interesting. So yes, you have already completed two research papers, I believe. So we'll now dig a little bit more about that. So can you please explain more about your first two topics in simple English? Like, can you break it down to the level that all our audience can understand?
Okay, so for my first research experiment, I looked into fruit flies who had Alzheimer's disease, and then
Okay.
I gave them the intermittent metabolic switching, which is a combination of fasting for 16 hours and eating for 8 hours, and also a high carbohydrate and low fat diet.
Okay.
And after I fed my flies this diet, I tested their memory, their behavior, and also their activity to see whether
Okay.
that would help with Alzheimer's disease. And then
Hmm.
lastly, I tested for neuroplasticity, which is the creation of neurons and new neuronal networks in the brain. So I tested to see
Okay.
if that could help increase brain function. And then for
How
my
about
second, huh? Yeah,
how about your second paper?
yeah. So for my second paper, I'm testing the gender in Alzheimer's. So how I did that is I used C. elegans, and then I used the female C. elegans in my
Hmm.
experiment, and then I
Okay.
gave elegans hormonal treatment because during menopause, which is usually when Alzheimer's starts to kick in, females' hormones levels drop rapidly. So that's why I supplemented hormones to my C. elegans to see if that would help make a difference. And then I tested for their memory and for
Okay.
their behavior.
All right, interesting, interesting. And I believe now you are in the process of writing your third research paper. How's your progress there? Can you explain more about it, perhaps?
Yeah, for sure. So I'm actually looking into depression therapies in
Okay.
correlation to Alzheimer's this year. And so how I'll be doing that is I'm just planning to modify this current depression therapy called brain simulation um, therapy, where they basically brain simulation
Mhm.
TMS therapy, where they basically shock you repeatedly to help with your depression. And I wanted to sort of
Mm.
modify that in regards to Alzheimer's disease. So I'm doing a modified TMS version to see if there could be any impact with Alzheimer's there. And yes, I'm currently working on my research. I haven't really analyzed my data yet, but I am collecting data as we go.
That's very impressive. So we'll now dig a little bit more about your completed research papers. So for your sophomore year project, for your first project, so what specific methods did you use to induce neuroplasticity in Drosophila melanogaster? And can you also explain what Drosophila melanogaster is? Because I'm pretty sure a lot of people here are not familiar with this term.
Yes, so Drosophila melanogaster is actually a common form of fly, the fruit fly, and I used fruit flies that lacked the amyloid precursor protein gene, which basically just led to amyloid beta plaques increasing in the flies and them having Alzheimer's disease, or them having the characteristics associated with Alzheimer's disease. And I induce neuroplasticity into my flies is using the IMS diet, intermittent metabolic switching, and how I did that is basically due to the combination of eating for very short periods of time and eating
Mm.
with low carbohydrates and high in fatty acids that can lead to a sort of G to K switch in the brain where your brain starts to rely less on glucose and more on fatty acids, and due to
Okay.
that switch can cause neuroplasticity.
Wow, that sounds so complicated. But all right, okay. And can you also describe the design and function of self-made teammates used in your research on Alzheimer's disease in fruit flies?
Yes, so I actually created the T maze where I have two sort of chambers on each side of the T maze, and then I have a middle chamber in the middle, and then the flies can
Okay.
cross over the chambers and into the middle. So when I slide the sliding door down, they get stuck in either chamber.
All right, okay. And what were like the key findings from your first project regarding the non-invasive preventative mechanism for Alzheimer's disease?
Yes. So there actually had not been a lot of research in regards to intermittent metabolic switching in Alzheimer's. There was no research at all. So I read an article about intermittent metabolic switching and neuroplasticity because I knew I wanted to incorporate neuroplasticity in Alzheimer's, and I saw that there might be a loose connection. So I decided to really go in and just test it. I didn't know if it would really work, but I hoped that it would, and in the end, it did work.
Hmm.
The memory of my slides did increase by about, I believe, 40%. And then
Okay.
my behavior of my flies was a little bit more negative than I expected. But that could be due to the changing brain chemistry in the fly brains. And then, last but not least, their activity did increase overall, and so did the neuroplasticity count in the flies. So that just showed that neuroplasticity was actually occurring, and that's what caused the memory to increase over time.
All right, okay. And yes, now we'll move on to your second research paper, your junior year project. So, how did you evaluate the effectiveness of hormonal therapy in reducing the incidence of Alzheimer's disease in C. elegans?
Yeah, so I basically just administered two different types of hormones, estrogen and progesterone, to my C. elegans in their food. And I chose these two hormones because progesterone hadn't really been tested in regards to Alzheimer's disease before. And then I tested estrogen because estrogen is the most common hormone in females that drop after Alzheimer's disease. And the reason why I tested these two was just to see if supplementing these hormones over time could help lead to increases in memory over time. So that's why I chose those.
Oh, okay, all right. And so, like, what specific hormonal therapies were like tested in your second project, and like, how about like their effects? Like, what are their effects?
So the effects of my two different hormonal therapies, pregnenolone and estrogen, um, it pregnenolone actually had the best effect
Okay.
as it showed that
Oh,
it only increased in memory significantly, but it also increased in A2M, which is a potential biomarker for the disease that I actually found while researching my experiment. And I found this that the amyloid two
okay.
precursor um, was actually a potential biomarker for the disease A2M, and I found that out because I tested A2M in an ELISA because there had been some concern about A2M before in regards to estrogen therapy, and I thought, let me test it to see if the concern is still there following my estrogen therapy, and I actually found that it can be a biomarker for the disease. In my experiment, and estrogen also had great results, but it was less as astonishing as pregnenolone, and also with estrogen, the behavior of the flies also decreased, whereas with pregnenolone, the behavior did not decrease.
Okay, and as you mentioned before, your junior year project like focused on like gender discrepancies. So, how did you measure this gender discrepancy in Alzheimer's disease in your junior year project, and what were your findings? Like, what was the conclusion there?
Yes, so I actually just separated the females and the males in the C. elegans
Yes.
separately, and those females had a much greater response to the treatments that I was giving them than the males. So I mainly focused my findings on the females because they showed a much greater reception to the therapies. And my conclusions basically stated that in the end, following the therapies, that progesterone was the best option, as it not only increased in memory therapy, but it decreased in the toxic biomarker in the C. elegans, and it also increased their behavior. So overall, progesterone was the best thing.
Oh, wait, can you say it again? What was the best?
Pregnenolone.
Okay, all right. Okay, sounds great. So yes, and for your senior project, what were the two model organisms you were using, and why were they chosen for studying subtype 4 of Alzheimer's disease?
Yes, so I'm actually studying subtype 4 in Alzheimer's disease, and I'm going to be using stem cell organoids, and I'm also going to be using fruit flies because fruit flies is a great way to memory in
Okay.
regards to Alzheimer's disease, which you can't really do with cells. And then I'm using
Mhm.
cells just to see if any of the therapies that I administer will be toxic to the actual cells inside our body because the choroid plexus is mainly a group of cells, and that's what I really wanted to test.
All right. Okay. Sounds great. And can you describe about your methodology that you're using for your senior year project, and how do they differ from previous research methods? I feel like you have already answered this before, but can you perhaps like reiterate?
Yes. So I'm going to be actually using cellular assays and tests this year because I haven't done that before. I haven't worked with cells yet. So that's going to be something completely new for me. Culturing cells and then testing them and testing their effects. And then in addition to that, I'm also going to be working with new therapies for Drosophila, including learning how to specifically shock them in specific ways and changing the voltage in order to really administer that TMS therapy that I was talking about. And I'm also going to be testing them for activity because that's going to be an aspect of depression that I also want to measure in
Mm,
Alzheimer's to see if that's a difference because activity
okay.
can be something that's really common in depression, and these therapies are supposed to help with activity. So I want to see if we can see that in Alzheimer's as well.
All right. All right. And how did your work on Alzheimer's disease research contribute to your induction into Sigma XI Research Honor Society?
Yeah, so actually, I presented my sophomore year project at their competition, the International Forum of Research Excellence, and I actually placed first there, and that led to them inducting me into their honor society when we went to Los Angeles to compete in their forum, and thanks to me winning, they inducted me into the society right there at the spot.
All right, thank you so much for your insightful answers. And like, yeah, as I was actually like interviewing you and asking a lot of questions about your research papers, I'm kind of curious though. Like, after you finish your third research paper, is there any other topic that you want to tackle in the future?
I'm probably going to continue with Alzheimer's disease, although I don't know
Mm,
in what sense in the future. I mean, I'd love to just continue researching it. Maybe in regards to mental health, because I always found mental health
okay.
sort of an interesting conundrum that I might want to look into.
All right. And before moving on to next section, like all of my GSU members are doing the research paper independently. So do you have any like tips on how to do this independent research in a better way? I believe you have a lot of tips to share with us.
Yeah, so I mean, I mainly will just suggest to find something that you're mainly passionate about because if you have a topic that you don't really like, you're not going to want to go to lab every day to research it. And finding a topic that you're really passionate about is a great way to start with your research journey. Additionally, you should always just never say I don't know in an experiment. Always
Okay.
try to back up with your prior knowledge. Research a lot before you go and compete, and just make sure that you know what you're talking about and that you can really explain it and show that passion that you have to the judges because the judges will just love to see that passion that you bring.
All right. Sounds great. And now we'll move on to your competition experience.